it's a delight to introduce Ken Van Golen. Comes to us from Delaware. He, prior to joining the faculty at Delaware, the faculty at Delaware, he developed an interest in inflammatory breast cancer, and I had no idea what this was really. It's a devastating tumor. Does not show up by mammography. It doesn't form a lump. Spreads, well, you'll tell us all this, spreads through tissues in a sheet, and is diagnosed usually 3B, stage 3B?
Stage 3B or stage 4.
How about that? So it's really a devastating disease. And when he was at Michigan, he was at Michigan prior to this, he developed his interest in inflammatory breast cancer and has been working on that since the early 2000s. Really, he's been one of the first people to do this. And as I said, what's worse about this tumor is that it's very hard to diagnose. It's usually diagnosed late. And he has numerous recognitions for his research. He organizes and leads conferences on inflammatory breast cancer. He's a member of the Inflammatory Breast Cancer Foundation Medical Advisory Board, and he chairs the research committee of the International Inflammatory Breast Cancer Consortium. So on a personal note, we were at dinner last night, and we were talking about, he comes from Michigan. He likes the Big Ten. He was delighted to come to a Big Ten university because of the sports competitions.
It wasn't the bar.
No. But he was at Michigan. They had a football team. A lot of ours are in jail. So we talked about something else after that. And then today, so his family came from the Netherlands 65 years ago.
60.
Pardon?
60 years.
Oh, wow, okay. And his coat, he wore, that's his dad's coat that he wore. 60 years old.
Yeah.
I'm 65. And Mike Leger says, well, who has the worst wear? Don't answer. Okay, Ken, you're going to talk to us about targeting in this awful tumor. And thank you so much for coming. I appreciate it.
Thank you for having me. It's truly a delight to be here. And let me clarify, it's good to be at a real Big Ten school. Not like your Nebraska, Rutgers, Maryland. Come on, really? That's not the Big Ten. No. So it's great, and thank you, Jim, for having me. Oops. Oh no. Okay, wait.
I guess the arrow.
There we go. Okay, so I was asked to put a disclosure slide. Very poorly paid academic scientist. I wish I had something to disclose. Unfortunately, I do not. I have a relationship with a company called AROG Pharmaceuticals, but all they do is give me free drugs. Well, experimental drugs. Not for personal use. So this is my outline for the seminar. I'm going to give you an introduction and a background in inflammatory breast cancer. One of my responsibilities at the University of Delaware is teaching. It's I get a hard salary for it, so I gladly teach, but I teach a basic introductory biology course to freshmen. And this year I have 165 freshmen in one of my classes, and I ask them every year, because I use inflammatory breast cancer as a case study to relate basic biological principles to. And I ask the class each time, has anyone ever heard of this disease? Typically in a class of 100 students, I get nobody's heard of it. This year I had one person that had heard of it, and it was because her grandmother was diagnosed with IBC. So I know, being at a cancer center or medical school, I'm sure there's people here that have heard of it, but I'm going to go through the signs, the symptoms, the biology, some of the molecular distinctions between IBC and non-IBC, or conventional breast cancer, however you want to call it. I'm going to lay out some of the current questions in IBC research, things that are really of interest to me and my laboratory. And then I'm going to go into the current research in IBC. I'm going to talk about two different projects that have gone on in my laboratory, or are going on in my laboratory. One is looking at RhoC GTPase and its interaction with AKT, which has been published. And then I'm going to talk about some newer stuff with platelet-derived growth factor receptor alpha, which actually one paper has been accepted, and we've got another manuscript that we're putting together. So I'm going to be showing you some fresh data, and then I'll go over the key points. So this disease is arguably the most aggressive and deadly form of epithelial breast cancer. Current SEER estimates say that this accounts for about 1 to 3% of breast cancers in the United States right now. However, the latest, greatest numbers, and I was just at an IBC meeting at MD Anderson this past weekend, the latest, greatest numbers say this accounts for 10% of breast cancer deaths in the United States. So reportedly, a small percentage of women have the disease, but account for a major amount of deaths in the United States right now. It's a phenotypically distinct form of locally advanced breast cancer. The main diagnosis is done clinically. What's taken into account, and actually I was part of the criteria for diagnosis back in the 2008 MD Anderson meeting, one of the very first international inflammatory breast cancer consortium meetings that ever occurred. Age is taken into consideration. Younger women tend to get this disease. Pregnant, lactating women. We have a number of high school age girls that die from this disease each year. And the youngest on record right now is a 9-year-old with inflammatory breast cancer. And there's several 12-year-olds that are in the literature as well. So there are some changes in the breast that occur. One thing that's most noticeable is the enlargement of the breast, nipple retraction, and soreness, and then of course these primary skin changes, the erythema, warmness to the touch, color changes from anywhere from a light pink, like what's shown here. I guess. Yes. Is this laser? Okay. No, I'll do it this way. It's fine. I'll do it this way. Anywhere from this light pink to this poor woman who the skin changes are a bright purple color. Okay. And then there's a texture, a unique texture to the skin, which is known as peau d'orange, or skin of an orange. It gets thickened and it gets a dimpling in the skin. So the onset of these changes, the enlargement in the breast, the primary skin changes, have been reported to occur as quickly as overnight. Women have reported going to bed completely normal, waking up the next morning, full-blown symptoms just like this. Okay, so extremely rapid onset. By definition, this disease is a T4D tumor. Okay, by definition, it's a T4D tumor. Oh, thank you. Okay, perfect. So there's a pathological diagnosis. So this is a lumpless cancer. The primary tumor presents as either sheets or cords within the breast. Therefore, it's almost impossible to detect by mammogram or by self-examination. One of the hallmarks pathologically are the tumor infiltrating into the ductal lymphatics. And so here's the epidermis. This is the dermis. Here are the ductal lymphatics, and here are tumor emboli invaded into the lymphatic system. And same thing here. Here's some emboli that have invaded into the lymphatic system. Okay, so at diagnosis, the tumor is either diagnosed as a stage 3B tumor, or in about 1/3 of women, they have obvious distant metastases. They're diagnosed as a stage 4 tumor. The 5 and 10-year disease-free survival rate is less than 45%. I believe the latest number is at 5 years is 42%. And at 10 years right now, I believe it's around 18%. So very, very guarded prognosis for these women. So what contributes to the lethality of IBC? First of all, because of the really unique symptoms that this disease shows, it's often misdiagnosed by primary care physicians and by general OBGYNs. So in greater than 90% of the cases, the women are thought to have mastitis. They're given rounds of antibiotics. Go 6 weeks with a round of antibiotics. The physician changes it, maybe gives them some steroids or something trying to clear up this disease, and it actually turns out to be a tumor. Okay, so they actually, because the progression of the disease is so quickly, they waste some very critical time being treated for an infection that actually doesn't exist. The disease itself is highly invasive and metastatic. You see it gets into the dermal lymphatics. It has access to the blood, to the circulation system, and it spreads throughout the body. In more than 90% of cases, 1 year after diagnosis, women will show up with recurrences, local recurrences as well as distant metastases. So you have these outgrowth of occult metastases that occur. And the sites of metastases vary between woman and woman. Pretty much I've been asking physicians for 20 years. I've been working on this disease since 1996, and I asked physicians for 20 years, what kills these women? They go, well, it kind of goes everywhere. And it does. So it goes to the bone, it goes to the brain, it goes to the liver, it goes to the lungs, it goes to the kidneys. It goes everywhere, and these women die of massive organ failure. So this is a molecularly distinct disease from other types of breast cancers. Our group, as well as several other groups, has spent quite a number of years looking at different things. And basically what I tell my students when they come into the laboratory, whatever you know molecularly about breast cancer, basically just forget about it or just turn it around and think oppositely. So there are some things that are common in both. For instance, they can be estrogen receptor and progesterone receptor expressors. They can express HER2. They can have loss of PTEN, increased EGFR, mutations in P53, just like in conventional breast cancers. In non-inflammatory breast cancers, though, you see things during progression like a decrease in the cell adhesion molecule E-cadherin. You see decreases in the plasma membrane proteins, KvanGolen and KvanGolen. You see mutations and changes in BRCA1 and 2. I'll talk about this. This is a unique gene that I cloned out when I was looking at IBC initially. In IBC, you see the opposite. You see increases in a GTPase, which I'm going to talk about during the talk, known as RhoC GTPase. You see overexpression of things like E-cadherin, KvanGolen, ALK, NFKB-related genes, PDGFRa alpha, and then loss of these unique things. No evidence for BRCA1 or 2 involvement in this type of cancer. And the cell type of origin can be, just like with non-inflammatory breast cancers, they can be luminal A, luminal B. That tends to be a very small proportion of IBCs, around less than 15% or so, the majority of which either are HER2 positive or basal, like triple negative types of tumors. So there are 3 questions that are really of interest to me, and they are of interest to the IBC community right now. So first of all, what is the origin of the disease? Believe it or not, there's evidence of clusters of this disease. So if you only think of this, like I said, it only accounts for 1 to 3% of breast cancers in the United States. But if you go to the state of Michigan, there's a tri-state area where there are 30 people within 10 miles of each other that were all diagnosed with IBC. And that includes a husband and wife and their dog. And their little dog, too. Yes, I'm not joking. I have a small amount of funding from a local IBC group in Delaware. The woman whose memorial this money was raised for actually lived in the neighborhood in back of me. She and her neighbor were diagnosed at the same time. Okay, so there's evidence for clusters. There's multiple clusters through the United States. There's a difference in ethnic distribution, so African American women tend to get at higher rates than Caucasian women, Latinos, American Indians, and Asians. Very low proportion of Asians get IBC. There's also distribution of rural versus urban. Most of these women that get it live in rural environments, not in urban environments. We can talk about this later. I'm not going to actually talk much about this in my talk, but we can talk about this later because it's absolutely fascinating to me, and I've got a small project going trying to look and see if there's some kind of infectious agent involved in this. So how does it metastasize? We know that it gets into the emboli, or the emboli get into the dermal lymphatics. What happens then? Okay. I'm personally interested in skin metastases. A large proportion of these women, about 25% after they've been treated, develop skin metastases, which is just a harbinger for death, essentially. They remain very, very sick for a number of years and then eventually pass away from overwhelming disease. And then, of course, the big thing, the biology is different, so can we find some specific targets for therapy? And that's going to be the main part of my talk today. So this is the very first paper that we published on IBC. This was published in 1999 in Clinical Cancer Research. This is about 3 years' worth of work. And this was the very first study ever to compare IBC with non-IBC. And this was while I was at the University of Michigan. And what we did in this study was we used the SUM-140. It was then the newly established SUM-149 inflammatory breast cancer cell line. And we compared it with immortalized human memory epithelial cells, as well as the lymphocytes that came from the patient that the SUM-149 was developed from. And we did a differential display technique at that time. That time we could have done gene chip analysis, but the gene chips were very small with very defined genes on them. And also at that time, people really didn't know what to do with the data. So we opted to do differential display at that time. And what we did was we came up with a list of genes that were either expressed by the normal cells and not by the tumor cells, or by the tumor cells and not by the normal cells. And then I took a number of patient samples, somewhere on the order of about 25 IBCs and 25 stage-matched non-IBCs. They were actually coded, so they were blinded, did in situ hybridizations looking at the different genes. 2 investigators categorized those in situ hybridizations. We had a pathologist go through, read them as well, confirmed what we were seeing. Then we broke the code, segregated them into IBC versus non-IBC, and then looked at what genes were expressed. And this is annotated a small list of some of the genes that we saw, and this is actually out of the paper itself. So the N's are the genes that were expressed in the normal tumors, or I'm sorry, in the normal cells versus the tumor cells, and the T's were expressed in the tumors but not in the normal. And then we have a comparison of IBC versus non-IBC, and then of course the statistics. 2 genes fell out of this. One was a novel gene, which I later cloned. I called it lost in inflammatory breast cancer, or LIBC. This was significantly lost in inflammatory breast cancers versus non-inflammatory breast cancers. The other gene that we found was called RhoC GTPase, which is a monomeric G protein, significantly expressed in about 90% of IBCs versus 38% of non-IBCs. And this is going to be more of the subject of as the talk goes on. So we also found a number of other genes that were at that time were not annotated. And a year or 2 ago, one of my graduate students and I, we wrote a book chapter on molecular mechanisms of IBC. And we decided to revisit some of the data and see if these genes were now annotated. They are. There's a whole host of new interesting genes, and if I ever get my wish where some old lady gives me $1 million or $2 million, and I will be able to have a lifetime worth of work looking at some of these new genes, because they look very, very interesting. Okay, so let's talk about Rho a little bit. So Rho stands for RAS homology. That's literally where the name comes from. It belongs to the Rho subfamily of the RAS superfamily of small GTPases. GTPases are enzymes that bind GTP to become active, and then they hydrolyze GTP as they signal. The Rho subfamily is overwhelmingly involved in actin cytoskeleton reorganization, although they do have other functions as well, and we'll talk about a couple of the other functions briefly in a moment. So this is the Rho family tree. In red here, this annotates where RhoC is found, and RhoC has two close homologs, RhoA, it's a very close homolog, and RhoB. And in fact, if we look at the sequence comparison between RhoC and RhoA, we see that they are very homologous to each other. They only differ by 15 amino acids. However, they are distinctly different when they're expressed in a cell. Those 15 amino acids make a world of difference. And I'm not going to go into why that is, but we can, if anyone wants to talk about it later, we can talk about it later. There are a number of reasons why these two proteins are very different. RhoC itself, if you express RhoC in a cell, actually if you overexpress RhoC in a cell, it will kill the cell. It is actually cytotoxic to the cell. RhoA is expressed in every single cell of the body, and it's highly expressed in every single cell of the body. And RhoA, if you did not express RhoA in the cells of your body, your cells would have no structure to them, because RhoA is what allows epithelial structure to occur, allows the stress fibers for your fibroblasts to crawl around, all of that. Okay? RhoC is actually only found in motile hematopoietic cells, so like dendritic cells, macrophages, things like that. Okay? If we take RhoC, and this is a number of years worth of work and millions of dollars of research, and it's all condensed into a single slide. If you take RhoC and you transfect it into an immortalized human memory epithelial cell, you go from this very nice epithelial morphology to this very kind of mesenchymal morphology with a leading edge of the cell. The actin cytoskeleton changes very, very drastically. You get migration and invasion. There are changes in growth patterns. Actually, the cells are able to grow that are expressing RhoC are able to grow under anchorage-independent conditions. They produce all kinds of crazy cytokines, including angiogenic cytokines, inflammatory cytokines, and it makes the cells mildly tumorigenic. So RhoC, just like all other monomeric G proteins, including RAS, the way that they're activated is through the GTPase cycle. So it's a transient off, on, off type of cycle. The GTPase is sequestered in the cytoplasm by a protein known as a GTPase dissociation inhibitor. It's prenylated in the C-terminal end upon stimulation, whether it's mechanical stimulation, stimulation due to receptor activation, heterotrimeric G protein activation, whatever. It's escorted up to the plasma membrane. The GDI dissociates. It's inserted into the inside of the plasma membrane. A guanine exchange factor then comes in. It recognizes the GDP. The guanine exchange factor actually abhors GDP. It plucks the GDP out of the binding pocket of the Rho protein and physically exchanges a GTP for it. That changes the conformation of the GTPase, allowing it to interact with a downstream effector. You get whatever the phenotype is. Then a GTPase activating protein comes in because the intrinsic hydrolysis capability of the Rho proteins is actually pretty low. Comes in, it increases the hydrolysis capabilities, it catalyzes hydrolysis. GTP is then hydrolyzed GDP. It becomes inactive and comes off the membrane, reassociating with the GDI. Okay? RhoC, there's an exception to this, and we'll talk about this in a few minutes. So we were interested in how RhoC was activated. I developed an interest in the protein AKT, otherwise known as protein kinase B. And one of the reasons why I became interested in it because we have some evidence that AKTs are actually overexpressed in nIBC. And there have been some literature, some very cryptic literature, I'll put it that way, that demonstrates that the Rho GTPases can be downstream effectors for AKT, so they can be phosphorylated by AKT. And this one paper demonstrated that RAC GTPase, which is a family member of Rho, could be phosphorylated by AKT, and actually its activity, so that whole cycling through the GTPase cycle, be inhibited by phosphorylation with AKT. So we asked the question if RhoC could act as a downstream substrate of AKT to mediate the invasive phenotype of IBC. And of course, I'll jump to the punchline, and I'll give you just a little bit of primer. Yes, of course it is, otherwise I wouldn't be talking about it, one. And two, it's inflammatory breast cancer, so it's the opposite of what they saw for RAC. Okay, so this is some microarray analysis that was done by our colleagues in Antwerp, Belgium. We've had a very, very productive, long-standing collaboration with a group in Antwerp. They have a number of patient samples on the order of some close to 300 IBC patient samples that are pre-treatment, and they have about twice as many matched on whatever level you want to talk about, stage, grade, cell type of origin, whatever, non-IBC patient samples that they compare with. And so they went through and they looked at PI3 kinase and AKT gene sets in IBC versus non-IBC. And we see here in red, these are all the genes that are overexpressed in IBC. In green, these are the non-IBCs. They went through and they segregated these into the different processes, and they found one process, process 4, that was significantly different in IBC versus non-IBC. These are genes that are overexpressed in IBC. And what this process is are the genes that are involved in the regulation of the actin cytoskeleton and cell migration. So we were pretty sure we were on the right track looking at AKT. So then the question is, which AKT? We know that all the AKT isoforms, all three isoforms, 1, 2, and 3, are overexpressed in IBC. There are reports that AKT2 is involved in non-inflammatory breast cancer cell migration. Of course, since it's involved in non-inflammatory breast cancer cell migration, would it be involved in inflammatory breast cancer? No. That's one of the things that we found. And what we found was that AKT1, 2, and 3 all overexpressed in the SUM149 IBC cells. This is an in vitro activation assay looking at stimulation of the cells. And then we compared them with the non-inflammatory breast cancer cells, MDA231, which is a triple negative. Non-inflammatory breast cancer cells, the 149s are also triple negative. And then the MB435s, which have been used for a lot of the AKT studies for non-inflammatory breast cancer cells. And then we threw an estrogen receptor positive cell line as well, MCF7s, just for comparison. So what we did was we used a pool of siRNAs against AKT1 and AKT2. In the majority of cell types and cancers, AKT1 is overwhelmingly involved in cell survival. It's not the case with inflammatory breast cancer cells. You can knock down AKT1 in the inflammatory breast cancer cells. You can stress them all you want, and they just keep growing. So we knock down AKT1 and AKT2 in these cells, and I'm sorry it's not projecting well. This is a transwell assay. These are the transwells. What you see is when we knock down AKT1, only in the case of the inflammatory breast cancer cells, these are still viable cells, the cells do not invade. There's a significant decrease in the cell's ability to invade in a transwell assay. In the non-inflammatory breast cancer cells, we see this when we knock down AKT2. So AKT1 inhibition lowers the SUM149, the IBC cell's ability to invade. AKT2 lowers the non-inflammatory breast cancer cell's ability to invade. Okay, so we asked, what's AKT3 doing? We would have included it in the other assay, but what we found was every time we inhibited AKT3, the inflammatory breast cancer cells died. So AKT3 is involved, this is percent apoptotic cells, is involved in IBC cell survival. And this data actually has been corroborated by a couple other laboratories. Frederica Robinson's laboratory at UVA has looked at this. She's looked at AKT3 in patient samples as well, and has corroborated this for us. Okay, so question is, does RhoC act as a substrate for AKT1? So this is an in vitro phosphorylation assay where we've taken a recombinant wild-type RhoC or a RhoC peptide that contains the putative AKT phosphorylation site. And this is a radioactive assay where we've added P32 and then an active AKT1 enzyme. As controls, we did the same thing with alkaline phosphatase. We also did a recombinant RhoC where we've replaced the serine at 73 with an alanine so it cannot be phosphorylated. Or we've used this peptide scrambled. And what we see is in the case of the wild-type RhoC, as well as the RhoC peptide with that putative AKT phosphorylation site, that we get a significant amount of those proteins phosphorylated. If we go to the cell lines and we do a pull down for RhoC and then a blot for phosphoserine, we see that in the SIM149s, we're able to pull down high amounts of phosphorylated RhoC. Next, what we did was we went and we knocked out endogenous RhoC out of the SIM149 cells, and then we re-expressed a wild-type RhoC, and we also re-expressed a RhoC containing the mutant that cannot be phosphorylated, then put them into an invasion assay, and we see that we get almost no invasion of these cells whatsoever. So this suggests that you need that phosphorylation at serine 73. We then did the opposite experiment where we've gone in and we've replaced RhoC with a phosphomimetic mutant, a vector alone phosphomimetic mutant, or a phosphomimetic mutant of RhoA GTPase. And I forgot to mention in the very first slide that if you put RhoA into breast cancer cells, or you look at the effect of RhoA, it has no phenotypic similarities to RhoC whatsoever. The cells basically just go, I'm here. So then we went through, we treated the cells with an siRNA, a scrambled siRNA, an siRNA to AKT1, to AKT2, or we treated them with a pan-Rho inhibitor, which is C3 exotransferase, and then we tested their ability to invade. To cut to the chase, only the RhoC phosphomimetic mutant was able to rescue the invasive phenotype of these cells. So this suggests that you have to have a phosphorylated RhoC at serine 73 at that putative AKT phosphorylation site. If you take the cells in which we expressed the S73A mutant, the mutant that cannot be phosphorylated, and you do a gene analysis, this again is done by our group, our collaborators in Antwerp, Belgium, the profile of the gene expression begins to resemble that of non-inflammatory breast cancer patients. So we switch from an inflammatory signature, and actually our collaborator Stephen Van Leer has demonstrated that there is a 73 gene signature. You go from that inflammatory gene signature to a non-inflammatory gene signature. So it looks more like a non-inflammatory breast cancer. So then the question is are the phosphorylation of serine 73, is that affecting anything within that GTPase cycle? So is it affecting Rho-GDI interactions? Is it affecting GTP hydrolysis, gap activity, anything like that? And this was about half of one of my students' master's projects. This was a huge amount of money, a huge amount of work, and the answer is there's no effect. I just feel obligated, since I paid a lot of money and she put a lot of time in, I feel very obligated to show this. So no effect. There's no effect on regulatory mechanisms that affect the GTPase cycle. Right, so in a project that's seemingly unrelated, several years ago, I developed an interest in KvanGolen-1. And KvanGolen-1 is a plasma membrane protein that's associated with cholesterol-rich regions of the plasma membrane, commonly known as lipid rafts and caveolae. Okay, KvanGolen-1 has this very unique, it oligomerizes and it has this very unique part to it right here, which is the KvanGolen scaffolding domain. This scaffolding domain associates with a number of signaling molecules. It can associate with receptors, it can associate with heterotrimeric G proteins, it can associate with monomeric G proteins such as RhoA and RhoC. We've shown it at least in pancreatic cancer. So I actually at the time I was looking at KvanGolen-1 in pancreatic cancer, and this was actually the very first time I was approached by the group in Antwerp, Belgium. This was our very first project together. And they came and they were interested in looking at KvanGolen-1 in inflammatory breast cancer. So we looked at cell lines and patient samples, and what we found was that KvanGolen-1 is overexpressed in nIBC patient samples. It's actually highly overexpressed. I'll show you that in just a moment. And the cell lines. Its overexpression is due to hypomethylation of the promoter. Okay, so you're losing methylation of the promoter, which is opposite to what you see in non-inflammatory breast cancers. And actually in non-inflammatory breast cancers, as progression occurs, you lose KvanGolen-1 expression, and that's due to hypermethylation of the promoter region. And then there was a distinct correlation of KvanGolen-1 and RhoC GTPase expression in human IBC samples. So we published that work, but we never went through and elucidated the function or the effect of KvanGolen-1 overexpression in IBC. This project actually has taken almost a decade to complete, because I would have a student that would start it and then go, I don't want to do this anymore, I'm bored, I want to do this project instead. And then I'd have another student start it, and they would take it a little bit farther, and then they would kind of fall off. I thought it was an interesting project myself, so I kind of put the whip to one of them, and she finally finished it. So this is the work of a recent graduate of mine, Madhura Joglekar. So one of the things that Madhura did was, one of the very first things she did was try to expand our insight into the expression of inflammatory breast cancer, expression of KvanGolen-1, not only in IBC, but also in intraductal carcinoma as well as normal breast. And she went through and she mined a database looking at the expression of KvanGolen-1. And what we find is this relative expression where normal breast epithelium has a level of KvanGolen-1 in it. Intraductal carcinomas, as they progress, as they become stage 3, stage 4, they lose KvanGolen-1 expression. But inflammatory breast cancer at stage 3, stage 4 actually expresses tons of KvanGolen-1, huge amounts. So to try to figure out what the effect of KvanGolen-1 overexpression was, we took 3 cell lines. We took immortalized human mammary epithelial cells. We took the MDA MB435s because a lot of work has been done on these, and KvanGolen work has been done on these cell lines, and then the SOM149 cells. We know that in the MDA435s and in human mammary epithelial cells, it's published, you have very nice expression of KvanGolen-1 on the plasma membrane associated with lipid rafts. We didn't know where it was in the inflammatory breast cancer cells, so we did confocal immunofluorescence for KvanGolen-1. We did immunogold labeling followed by transmission electron microscopy, and then a cell fractionation, fractionating out cholesterol-rich membranes. And what we find is KvanGolen-1 is highly expressed on the plasma membrane, but there's also intracellular stores of it as well. Not unusual to see in inflammatory breast. Exactly the same. We see exactly the same thing with E-cadherin, which is a cell surface protein. Tons of it in the cytoplasm as well. Okay, so what we wanted to do was knock down KvanGolen-1, but not completely knock it out. What we wanted to do was just knock it down to normal breast levels. And that's what we did here. We knocked down the message and got the protein levels down to what you would see in the human mammary epithelial cells. We looked for any changes in cell proliferation, cell viability, that kind of didn't see any effects with that. However, we saw profound effects in the cell's ability to invade. So if we knock down KvanGolen with the siRNA, we see a significant decrease in the cell's ability to invade. If we use methyl beta cyclodextrin, which chelates cholesterol out of the membrane, dispersing KvanGolen through the membrane, so you lose those lipid rafts, we see a significant decrease in the cell's ability to invade. If we introduce a KvanGolen scaffolding domain that acts as a bait to pull away signaling molecules, we also get a significant decrease in the cell's ability to invade. So we look to see if KvanGolen was associated with RhoC GTPase, and we do see them co-localizing with each other in the leading edge of a motile cell. By biochemical methods, we could not demonstrate that they were interacting with each other physically. However, we did show that in pancreatic cancer, so it was a little bit of a surprise. However, we did demonstrate that when you knock down KvanGolen or you introduce this KvanGolen scaffolding domain, you get about a 40% decrease in RhoC activation. So the GTP level of RhoC goes down by about 40%. So there's still 50%, more than 50% of RhoC that's active in these cells, but you still get these tremendous decreases in cellular invasion. So we ask the question, is this affecting AKT1 activation? There's evidence in the literature that shows that active KvanGolen is required for activation of PI3 kinase. PI3 kinase in turn activates AKT1. And sure enough, if we look at the KvanGolen scaffolding domain or the knockdown of KvanGolen, we get a pretty significant decrease in active AKT1 in the inflammatory breast cancer cells. And this is the ratio of active to total AKT1. You can see that's pretty significant. We do the pull down of RhoC with immunoblot for phosphoserine. We see that, yes, indeed, phosphorylation of RhoC is decreased in these cells as well. So then we went through, we made these mutants. Why not use them in this study as well? We introduced RhoA with the phosphomimetic mutant, RhoC with the mutant that cannot be phosphorylated by AKT, or RhoC with the phosphomimetic mutant. In each case, when we knock down KvanGolen, we use a pharmacologic inhibitor to AKT, or we use a PI3 kinase inhibitor. In each case, it's only the phosphomimetic mutant of RhoC that can rescue the invasive phenotype. Okay, so out of this study, or concurrently with this study, going through this gene set of all these different genes that were overexpressed, we started kind of looking at some of these, and there are a couple of them that are pretty interesting. This one in particular was chosen by one of my students, actually probably one of the most talented PhD students that I had, Heather Lehman. And this gene is platelet-derived growth factor receptor alpha. Data from Stephen Van Leer in Antwerp demonstrates that if we look at PDGFR signaling, specifically if what he did was he took a training set. I have to talk bioinformatics now. See if I can do it. I don't do it as well as Stephen does. He took a training set. What he did was he took gastrointestinal stromal tumors that harbored a constitutively active mutation and used it as a training set to query the inflammatory breast cancer patient samples and non-inflammatory breast cancer patient samples, and then looked at what percentage of those were expressed a constitutively active PDGFR alpha. And then these are whisker plots of the segregated data. So what we see is in non-stage matched, stage matched, ERB2 or HER2 overexpressors, luminal A's, luminal B's, in each case, there's a significantly higher portion of inflammatory breast cancer cells that express an activated PDGFR alpha signature. Only when you're looking at the basal type, the triple negatives, are they about the same. Okay, and we see that quite often too. A lot of times what you see in triple negative breast cancers, you'll see similar results in inflammatory breast cancers. So this is work that was done by Francois Bertucci in Marseille, France, also a collaborator of ours. And what he did was he went through patient samples and looked at the metastasis-free survival rates of patients that had this activated PDGFR alpha signature versus those that did not have an activated PDGFR alpha signature. And what we see is those patients that have the activated PDGFR have a significantly decreased metastasis-free survival rate. So their overall, it's a surrogate for overall disease-free survival. So these patients actually live less. So went through, looked at two different inflammatory breast cancer cell lines, the SOM149 line as well as the KPL4, which is actually derived from a skin metastasis of inflammatory breast cancer, and then compared it with the triple-negative MDA MB231s. What we see is, in the case of the inflammatory breast cancer cell lines, we have a constitutively active PDGFR alpha in these cells. Oh, this is one of those slides. Okay, so we asked the question, could we inhibit PDGFR alpha with a tyrosine kinase inhibitor? We started with imatinib, commonly known as Gleevec, which not only targets both PDGFR alpha and beta, but also c-KIT and BCR-ABL. It binds to the inactive conformation of PDGFR alpha, and it's a current treatment in a number of other tumors, acute myelogenous leukemia, gastrointestinal stromal tumors, glioblastoma multiforme. The thing that was attractive about this, it's an oral route of administration, high bioavailability. Then we also got hold of another drug. This is an experimental drug from AROD Pharmaceuticals, which is known as Crenolanib. This is the drug that they give me for free. It's specific against PDGFR alpha. It does target beta as well, but it specifically was designed to target alpha. It binds to the active conformation. So what they did was they designed this drug actually for gastrointestinal stromal tumors that harbor an activating mutation. It's highly potent. There are a number of clinical trials in progress, particularly those in gastrointestinal stromal tumors. So this is simple growth assays. Essentially what we demonstrate is the MDA231s, they're affected by both imatinib and Crenolanib. The inflammatory breast cancer cells completely refractory to imatinib. You can be putting water into the cultures with these. But Crenolanib's highly effective, and we get significant decrease in cell growth at 0.5 or 0.05 micromolar concentration. So very, very effective. Now the problem is inflammatory breast cancers don't grow in monolayer anaplastic. The real concern is the emboli that are in the dermal lymphatics. Several years ago, we published this in vitro model of growing emboli in culture. So this is matrigel, so these are mammospheres. And you see if you take MCF10As, which are basically mammary epithelial cells, non-inflammatory breast cancer cells or inflammatory breast cancer cells, you're able to grow mammospheres in matrigel. The environment of matrigel is not the same as the dermal lymphatics. So we have actually a suspension culture system that mimics the mechanical properties of the dermal lymphatics. And you can see only the inflammatory breast cancer cells grow in this system. We can either do it with a supplement of polyethylene glycol or more biologically relevant hyaluronic acid, since the lymphatics recycle hyaluronic acid. And if we grow these emboli and then we treat them with imatinib, essentially what we get is we treat with imatinib, we get perfect formation of emboli. However, when we treat with Crenolanib, we get an inhibition of the emboli formation. So essentially we can do it two ways. We can put it in the system to begin with. The emboli never form, or we can grow emboli, add the Crenolanib, and the entire emboli goes like this and just comes apart. But the proof is what happens when you put this into an animal. Okay, so We took nude mice. We injected the SUM-149s into the nude mice into the mammary fat pad orthotopically. We grew the tumors to 200 cubic millimeters. We then randomized the mice into 2 groups. We took Alzette micropumps, put either DMSO vehicle control in one set or Crenolanib in another set, implanted them into the mice, and then followed the mice for the 10-day life of the micropump. And you can see that in that 10 days, this is a typical growth pattern for SUM-149s in the mammary fat pad. These cells grew, or the tumors grew, almost 4 times in size, while the tumors in the Crenolanib-treated mice remained static. Interestingly, after the pumps were depleted, these tumors started growing. So this is a tumor-static compound. So the very last thing that I'm going to show you, and this is very brand new data. Okay, so we asked the question whether or not Crenolanib treatment could act as a chemosensitizer. And so this is all done in vitro, and we've done several different chemotherapeutic options, mostly the chemotherapeutic options that are used for inflammatory breast cancer patients. So in this case, what we've done is we've looked at docetaxel. This is docetaxel survival curve on the SUM-149s. You can see with different amounts of chemotherapeutic agent, and only the higher 2 concentrations, the 2.5 nanomolar and 5 nanomolar, actually achieve significance. However, if we combine docetaxel with 1 micromolar Crenolanib, which is very achievable in vivo, we get the same type of killing as we do with the higher amount of docetaxel alone. So we're able to use 5 times less chemotherapeutic agent to achieve the same amount of killing. And then this is a comparison of doxorubicin as well as paclitaxel. And basically what it comes down to is it mirrors what we see up here, is we're able to use significantly less chemotherapeutic agent to achieve the same amount of killing when we combine it with Crenolanib. So right now we're trying to get this study funded so that we could take this to do more preclinical work in the animals and work up a bunch of animals before this hopefully goes to the clinic for a clinical trial. And we've had some interest from the Dana-Farber group to look at this actually in patients. So I'll just end with some key points. So I hope I've convinced you that IBC is a phenotypically and molecularly distinct form of breast cancer. I hope I've shown you that RhoC drives the IBC invasive and metastatic phenotype, and that AKT1 regulates RhoC-driven invasion through phosphorylation at that putative serine 73 site. AKT3 appears to regulate IBC cell survival. KvanGolen appears to have a role in AKT activation. And PDGFRa appears to be a prognostic factor in IBC, which also, with the use of Crenolanib, could be a potential therapeutic target, and Crenolanib can be a potential therapy in IBC. I'd like to acknowledge my lab members. All these people did the work, and I present it. It's all my ideas, right? My former postdoctoral mentor, I always have to give her a shout because that very first slide that I said is a lot of work and a lot of money, that was all funded by her, Sophia Mariver at the University of Michigan. We have a very nice, long-standing collaboration with the Translational Cancer Research Unit in Antwerp, Belgium, specifically with Stephen Van Leer. AROG Pharmaceuticals gives me all the drugs I want. And then part of the International IBC Consortium, Francois Bertucci and Pascal Finetti, as well as Nato Ueno and MD Anderson, have worked with us looking at the patient samples and the metastasis-free survival and whatnot. Funding was with the DoD Breast Cancer Research Program. We've had some luck with them in the past, not so much in the present. The Deidre DiOrlo Memorial IBC Gift Fund. This is the woman who I said lived in the neighborhood behind me who died of IBC. and the IBC Foundation Network, which has actually been funding our most recent work on skin metastasis, which is really, really exciting. And maybe in a year or so, I'll have some more, get another invitation back to talk about skin metastases. So thank you very much. I appreciate your attention. I'll try to answer whatever questions I can.
I just had two things. One thing is the signaling and the phenotype of this disease also sound a whole lot like LAM or lymphangioliomyomatosis, which tumors form in the lung and they're mesenchymal from the uterus before there's even a uterine tumor.
Yeah.
And it's also a mutation upstream of AKT.
Oh, yeah, yeah.
Which turns off the inhibitor of mTOR signaling. So there might be some commonalities. Maybe they have PDGF or something like that. That might be cool.
Cool.
The other thing that is kind of weird to me is it's almost like you have Rho upstream of AKT.
Mm-hmm.
It activates AKT.
No, I would say Rho's downstream of AKT.
Okay, so
Rho's a
in my
substrate.
head-
RhoC is a substrate for
Yes,
AKT.
of course it is, but typically in signaling pathways, you want to put the G protein above.
Right.
And so maybe your mutation doesn't affect the G protein activity of it because that's not the activity.
Right.
You want to go downstream of that. So have you ever thought that RhoC is actually has a new activity?
Oh, absolutely.
As a transcriptional partner at the PDGF gene and has shifted.
Absolutely.
It's probably down in there.
Yeah, absolutely. Absolutely.
So then it wouldn't surprise you that it doesn't affect the GTPase because that's not what you're.
Honestly, in this disease, nothing surprises me.
Have you ever looked at the nuclear pore?
No.
Okay.
No.
Good question. So what kicks off AKT? I mean, what's very upstream? Is there a cell surface kinase? Or
So
have you all of it? Some
two
kinase has to
two things that we think kick off AKT. So I'm trying to draw a link between PDGFRa and AKT. Okay, so that's one. The other thing is mechanical forces. So I had last year I had two very, very talented medical students come from the Netherlands to work in my lab. One actually is related to me. It's my cousin's daughter. Another one who actually we convinced that she needs to do an MD PhD did a very, very large project on TRP channels. And one of the things that we see is that mechanical forces, because what you have to remember is these cells, if you get away from the primary tumor, which is a sheet or a cord in the breast, everybody focuses on and thinks is the key are those emboli in the dermal lymphatics, because those emboli are what are spreading through the body. Okay, there's constant oscillatory shear forces going because that's how lymphatic fluid moves, right? And so we think that the mechanical forces are what are tripping off the AKT.
Is there a fibrotic component to this tumor? I mean,
Is there a fibrotic?
increased collagen in the area or roadways?
No,
Things can migrate?
I'm trying to because there were a lot of talks about that this past weekend. No, the stroma changes. The stroma changes, but I can't remember what the actual changes were. That was something we wanted to look at a while back
There's
as
a
well.
lot of interest in non-inflammatory breast cancer, which is reorganization and changing
Right.
the intensity of the signals and the directionality of the movement. So
Right.
maybe something as well to look into. Are there other questions?
IBC cells invade lymphatics, and that's what causes the clinical presentation.
Correct.
Do they also invade venous structures?
So the idea is that they get, not directly, but the idea is that they get to the blood via the lymphatics.
Yeah. Well, they go to the lymphatics, but they don't immediately invade venous structures.
Correct. Yeah, correct. At least not that we've, never say never. Right? Never say never.
Several phosphorylation sites in AKT carried out by distinct kinases, and they often have different functionality.
Right.
I was wondering if you have some indication that a particular site may be more active?
Yeah,
I'm
so-
curious about because one of the sites is phosphorylated by protein kinase CK2,
Right.
and apparently creates a hyperactive AKT. Whether that has an impact in the
Right. So we actually interrogated multiple sites when we looked at the phosphorylated AKT. And be honest with you, I'm blanking on which site we ended up settling on. But there was one that was common that we found in the 149s that was common with other lines, other IBC lines, as well as what we saw in patient samples. And you'll have to forgive me because I'm blanking on. This was a 2012 study, so new things have populated my mind. But you're absolutely correct.
I think we have another meeting that we need to have. But anyways, thank you.
Thank you.